cross the blood-brain barrier. So what happens? In the brain, ammonia is taken up by alpha-ketoglutarate to form glutamate, which in turn form glutamine. So what is happening? Your excess ammonia, this is crossing the blood-brain barrier. So it reaches the brain. Now in the brain, what is happening? This ammonia is taken up by alpha-ketoglutarate. So this is accepted by alpha-ketoglutarate, and by accepting ammonia, alpha-ketoglutarate will form what? Will form glutamate. So your glutamate level in the brain is increasing. And this glutamate, this by accepting ammonia will form glutamine. So your glutamine level is also increasing in the brain. So in the brain, ammonia is taken up by alpha-ketoglutarate to form glutamate, which in turns by accepting ammonia forms glutamine. Now glutamine is an osmotically active compound. That means it has the property of attracting water. This causes what? Cerebral edema. Okay. And another thing, excess glutamate on decarboxylation will produce GABA. We've already seen that amino acids on decarboxylation, as I told you in our initial introductory chapter, that they produce biologically important amines. So it produces on decarboxylation a biologically important amine, that is GABA. What is GABA? It is an inhibitory neurotransmitter. So it causes what? CNS depression. So when your glutamate level in the blood increases, it gets decarboxylated, producing what? GABA, which is an inhibitory neurotransmitter, thus leading to CNS depression. Also, as your alpha-ketoglutarate is being depleted, so what is happening? Your alpha-ketoglutarate is a very important substrate for TCA. So your TCA is getting affected, and in turn, your ATP production is decreased. So there is decreased ATP production, and also ATP is being used by glutamine synthetase. Okay, glutamine synthetase is the enzyme which converts your glutamate into glutamine. So that reaction is also increased. So there also your ATP is being used. So in turn, there is additional ATP depletion. So there's further depletion of ATP. So what all is happening in the brain? A quick summarization, that ammonia is increased, your alpha is affected, so your ATP is decreased. Also, your glutamate is increased, therefore your GABA is increased, and GABA causes what? CNS depression. Okay. And also your glutamine is increased, and glutamine has the property of attracting water molecules. So what is happening? It causes cerebral edema. Okay. So this is how we say that ammonia is toxic to the body. As you can see, it is causing encephalopathy. It is affecting the structure and function of the brain. And what is GABA? The full form of GABA is gamma aminobutyric acid. It is the primary inhibitory neurotransmitter in the brain. So GABA is gamma aminobutyric acid. Okay. This is your okay. So that was briefly about toxicity of ammonia. So now we know that why is it so important to handle ammonia carefully until it is excreted from the body. Now let's begin with urea cycle. So urea cycle is also known as Krebs-Henseleit cycle. Okay, it is also known via the name of ornithine cycle. Why is it known as ornithine cycle? Because we'll see that ornithine is regenerated. Because ornithine is regenerated. Okay. And it is also known as urea bicycle. Why is it known as urea bicycle? Because TCA and urea cycle are linked together. So your urea cycle is linked with TCA. Therefore known as urea bicycle. We'll see as we proceed that in TCA, what is formed? TCA aspartate is formed. And that aspartate which is formed in TCA, it enters into urea cycle. And in urea cycle, what is formed? Fumarate is formed. And that fumarate which is formed in urea cycle, it enters TCA. So this is how your TCA and urea cycle are linked together, and thus it is also known as urea bicycle. Again, there's some lag, audio video lag. I think there's some network issue. Okay, anyway, so let's proceed. Okay, so now before we begin, let's have a look at the structure of urea. Your urea, it consists of 2 nitrogen and 1 carbon. As you can see, these are the 2 nitrogens and 1 carbon. So this nitrogen, one of the nitrogens, it is coming from ammonium ion. And the other nitrogen, it is contributed by aspartate. And this carbon, it is coming from respiratory carbon dioxide. Okay, so this is the structure of urea consisting of 2 nitrogen atoms and 1 carbon atom. The 2 nitrogens, they are donated by ammonium ion and your aspartate. Where is the urea cycle taking place? It takes place only in liver, and it is both cytoplasmic and mitochondria. Let's see. So you can get in the MCQs that urea cycle occurs in, so you know it occurs in liver. Uh, Also for the organelles and whether the cycle is cytoplasmic and the organs where it is occurring, you can also prepare a chart for this as well. You know, all where you have on one side, you have all your cycles like glycolysis, where it is occurring, gluconeogenesis, glycogen synthesis, urea cycle, TCA, everything, and the site that where it is it taking place. So a day before exam, you can just skim through it and it becomes very handy for you at the last moment. Okay, nitrogen atoms of urea, they are contributed by, so they are contributed by, we've just seen, ammonium and aspartate. Now it also asked, true about urea cycle, nitrogen of the urea comes from alanine and ammonia. It doesn't. Uses ATP during conversion of, okay, we'll get back to this later because we haven't studied the steps. Okay. Now, moving forward to the reactions of urea cycle. Now let's see what is happening. The one in pink, these pink boxes, this indicates that this is taking place in the mitochondria. Okay, so the pink area is indicative of mitochondria. Now let's see the reactions. So what is happening in the mitochondria? Your ammonia, it combines with carbon dioxide and water to form what? Your carbamoyl phosphate. So carbamoyl phosphate is being formed. The enzyme that is used here is carbamoyl phosphate synthetase. And this is the rate limiting enzyme. Okay, this is the rate limiting enzyme, and you can see that 2 ATPs are used in this reaction and they are changing into 2 ADPs. So, which is equivalent to approximately 2 high energy phosphates. Okay. So, in the first reaction, which is taking place in the mitochondria, what is happening? Your carbamoyl phosphate is being formed via the rate limiting enzyme of the reaction, that is carbamoyl phosphate synthetase. Now this carbamoyl phosphate synthetase, it combines with ornithine to form what? To form citrulline. Okay, this is also taking place in the mitochondria. Now this citrulline comes out into the cytoplasm and it combines with aspartic acid. So your citrulline combines with aspartic acid to form arginosuccinate, and this is catalyzed by the reaction ornithine transcarbamylase. Okay, so this here is catalyzed by ornithine transcarbamylase. No, this one here. So carbamoyl phosphate, I'll repeat it once again, okay? What is happening in the mitochondria? So ammonia and carbon dioxide, they combine together to form carbamoyl phosphate, which is via the enzyme carbamoyl phosphate synthetase. Now this carbamoyl phosphate, it combines with ornithine, and this is brought about by the enzyme ornithine transcarbamylase to form citrulline. Okay. Now this citrulline is combining with aspartic acid to form arginosuccinate. For this, the enzyme is arginosuccinate synthetase. Okay. So the enzyme here is arginosuccinate synthetase. In this reaction, you can see one ATP is being used, but it is changing into AMP. Okay, so one ATP, directly you're using one ATP, but it changes into AMP. So it is equivalent to 2 being consumed. And where is aspartic acid coming from? This aspartic acid, it comes from taking place in the mitochondria. So this aspartate comes via a transporter from the mitochondria into the cytoplasm. So name of this transporter is citrin. Okay, your aspartate is being formed in the TCA cycle, and your TCA cycle is taking place in the mitochondria. So this aspartate has to be transported from the mitochondria into the cytoplasm. So for this, there is a transporter known as citrin. Okay, now your arginosuccinate has been formed. Now this arginosuccinate via the enzyme arginosuccinate lyase is split into arginine and fumaric acid. So you see, this is a lyase enzyme. So lyase enzyme, they break covalent bond without adding water. You can see. So your arginosuccinate is split into arginine and fumaric acid, and this is brought about by an enzyme that is arginosuccinate lyase. Now, this arginine is acted upon by the enzyme arginase. Arginase enzyme to form what? Your urea and ornithine. Okay, now this arginase enzyme, this is a hydrolase. Okay, arginase enzyme is a hydrolase. So what are hydrolases? They break covalent bond by adding water. You can see water is being added in this reaction. And what has been regenerated? Ornithine has been regenerated, as we've already stated in the beginning of the lecture, that urea cycle is also known as ornithine cycle. So you see how ornithine has been regenerated. Now this ornithine will move into the mitochondria from the cytoplasm and will again combine with another carbamoyl phosphate, and the cycle will continue. So these were the reactions of urea cycle. I'll quickly summarize them again, okay, so that it's clear. And also, as you are taking the lecture, you're also memorizing things along with me. So what is happening in the mitochondria, as we've said, that urea cycle is partly mitochondrial and partly cytoplasmic. So the initial two steps of the urea cycle are taking place in the mitochondria. In the mitochondria, what is happening? So ammonia and carbon dioxide, they combine together to form carbomoyl phosphate. That carbomoyl phosphate then combines with ornithine to form citrulline. This citrulline comes into the cytoplasm, and in the cytoplasm, it combines with aspartate. Aspartate is coming from the TCA cycle. Okay. Now, citrulline and aspartate combine together to form arginosuccinate. Now, this arginosuccinate is split into arginine and fumarate. And this fumarate that is being formed, this is entering into TCA cycle. Okay. Now, the arginine is acted upon by the enzyme arginase, which is a hydrolase, which acts by adding water and breaking covalent bond. So what is split? It is split into urea and ornithine. So your urea is excreted and your ornithine is again transported back into the mitochondria to again combine with another carbomoyl phosphate. Okay, so that was about the reactions of urea cycle. Now moving forward. Okay, let's take a few MCQs. Which enzymes are part of urea cycle? Now let's see, ornithine transcarbamylase. Yes, this is very much part. Asparaginase, no, this is not. Glutamate synthase, no, this is not. Arginosuccinase, yes, this is. Okay. Now, which of the following is true in relation of urea cycle? It says first two steps in cytoplasm. No, this is not right. First two steps in mitochondria. Yes, this is correct. Defect of enzyme of any step can cause deficiency diseases. Yes, this is also correct. Urea is formed by ammonia, glutamic acid, and carbon dioxide. No, this is not true. It's glutamic acid. No, it's formed by aspartate. Citrulline is formed by combination of carbomoyl phosphate and ornithine. Yes, this is correct. Okay. Now let's have a look at the energetics of urea cycle. CPS1, that is carbomoyl phosphate synthetase 1, it uses 2 ATP, which is equivalent to 2 high energy phosphate. You can see here 2 ATP, they were being changed into 2 ADP. So 2 high energy phosphates were being used. Whereas arginosuccinate synthetase, it was using 1 ATP, but that 1 ATP was changing into AMP. So it was using 1 ATP, but 2 equivalents of 2 high energy phosphate, which is equivalent to 2 high energy phosphates. So you see 3 ATPs are being used directly, but 4 high energy phosphates are being used, which is equivalent to 4 high energy phosphates. Okay. Now regulation of urea cycle. High protein diet increases urea cycle enzyme synthesis. Okay. NAG is an allosteric activator of CPS1. So this first step of urea cycle, you can see here. This is activated by NAG. What is NAG? NAG is N-acetyl glutamate. This is NAG, and this is an obligate allosteric activator of the first step of urea cycle. What does it mean by obligate allosteric activator? That is, it is compulsory for it to be present. Okay, so the first reaction will take place only in the presence of NAG, that is N-acetyl glutamate. Okay. And then compartmentalization. That is the first and the second reaction are taking place in the mitochondria and rest in the cytoplasm. So this compartmentalization is also providing an extra layer of regulation to the urea cycle. Now moving on to the disorders of urea cycle. These are important. So if there is any defect or deficiency or block in the first enzyme, that is carbonyl phosphate synthetase, okay, if the block is here at this level, it causes what? Hyperammonemia, and it is of type 1 type. That means there has to be a type 2 as well. So your type 2 is caused when the block is in this level, that is ornithine transcarbamylase. Okay, when your ornithine transcarbamylase, if the block is here at this level, what is being caused? Hyperammonemia type 2. Okay, let me just do a segregation of this here. This yellow line that I have drawn, this indicates compartmentalization. Okay, so this is the mitochondrial line. Basically, I'm trying to show this side is mitochondria and this is your cytoplasm. Okay, and I told you that there was a transporter, citrin transporter. Okay, this citrin, this was the transporter which was transporting your aspartate. Okay, aspartate was coming from the mitochondria into the cytoplasm via the citrin transporter. Now let's see what is happening if the block is in at the next level, that is the enzyme arginosuccinate synthetase. Okay, this is synthetase. Okay, arginosuccinate synthetase. If the block is here, what will it cause? It will cause citrullinemia type 1. So there has to be a type 2. So the type 2 is when the block is in or there is defective transporter is defective. Then it causes citrullinemia type 2. Okay. So when the block is in the first step, that is your hyperammonemia type 1. If the second step, that is formation of citrulline is affected, then it is hyperammonemia type 2. Then if the block is in arginosuccinate synthetase, it causes citrullinemia type 1, and if citrin transporter is affected, then it causes citrullinemia type 2. Now, if the block is in arginosuccinate lyase, okay, this is lyase, then it causes was arginosuccinic acid urea. Okay. Now, if your arginase, the block is here, it's affected, then it causes what? Arginemia. Now, if your ornithine transporter, your ornithine is going from the cytoplasm into the mitochondria via a transporter. Now, if this ornithine transporter is defective, then you have HHH, that is triple H syndrome. Okay, so I've shown you in a circular in the cycle itself that at what levels if the block is, so what will be caused. Now, if the defect is in the first two enzymes, okay, that is in carbonyl phosphate synthetase or in ornithine transcarbamylase, okay, that is in CPS1 or in OTC. If the defect is in these two, that is the first two enzymes, then what is accumulating? More of ammonia accumulates, okay? More of ammonia accumulates. Therefore, it is known as hyperammonemia. But if the block is in the later enzymes coming in the cycle, then your name of the diseases is on the respective intermediates that are being accumulated. Okay. Now let's see in the tabular form your hyperammonemia type 1 by CPS1. Okay, as we've seen in the cycle here. Type 2, it is caused by ornithine transcarbamylase. Then hyperornithemia by defective ornithine transporter protein. Okay. Citrullinemia by arginosuccinate. Arginosuccinic aciduria by arginosuccinate lyase. And your hyperarginemia when the defect is in enzyme arginase. Okay. Now it says, let's take an MCQ. In which of the following condition there is increased level of ammonia in blood? So ornithine transcarbamylase, yes. Galactosemia, histidinemia, phenylketonuria, no. So the answer will be ornithine transcarbamylase deficiency. As we have seen, it causes hyperammonemia type 2. Now let's study a peculiar thing about hyperammonemia type 2. That is its distinguishing factors. It says that your ornithine transcarbamylase is defective, leading to accumulation of carbamyl phosphate. Okay, so carbamyl phosphate will accumulate. Let's see how. Because in the first step, your ammonia was combining with carbon dioxide to form what? Carbamyl phosphate. The first enzyme was present, so your carbamyl phosphate is formed. Now your carbamyl phosphate was supposed to combine with ornithine to form what? Citrulline. But this here is not taking place. Okay, so the block is at this level. So what is happening? Your carbamyl phosphate is formed and it accumulates since it cannot combine with ornithine to form citrulline. And this was taking place in the mitochondria. Okay, this was mitochondria. So now what happens? Your carbamyl phosphate, it comes out of the mitochondria into the cytoplasm. So your carbamyl phosphate comes out and it goes into pyrimidine synthesis. Okay, and in pyrimidine synthesis, there is an important intermediate being formed known as orotate. Now, this orotate then forms uracil, from which are formed pyrimidine, cytosine, and thymine. So, what is happening is your pyrimidines are increasing. Okay. Now, this orotate that is being formed, it is insoluble in water. This is insoluble in water. So it forms orotate crystals are being formed. Okay. So that is the peculiarity of type 2 hyperammonemia. Let's see what it says. Carbamyl phosphate comes out of the cytoplasm from the mitochondria and enters into pyrimidine synthesis. Okay, because it is accumulating, so it comes out and enters into pyrimidine synthesis. Now this orotate, an important intermediate in pyrimidine synthesis, is formed, which forms uracil, which ultimately forms cytosine and thymine. Now, orotic acid is insoluble in water and forms orotate crystals, causing orotic acid urea. Okay, so your orotate crystals are accumulating, leading to orotic acid urea. It forms pink gravels in the urine. So, what are the distinguishing features of hyperammonemia type 2? Let's see. Features of hyperammonemia type 2. Firstly, what will you see? Orotic acid urea. How? Because your orotate crystals, they are insoluble in water, so they accumulate leading to orotic acid urea. And second, there is excretion of pyrimidines in urine. So these are the two distinguishing factors of hyperammonemia type 2. Okay. Now, arginosuccinic aciduria. Here, what is the distinguishing feature? That is trichorexis nodosa. You have dry, brittle hair. You can have small nodules are present in the hair. Okay, it will look something like this. Small nodules present in hair. Okay. Now, moving on to HH8 syndrome. HH8 syndrome, it is due to defect in ornithine transporter. So your ornithine transporter is defective. Manifestations include hyperammonemia, hyperornithemia, and homocitrullinemia, yeah, urea. So this is where your H, and H comes from. Okay, this HH8 syndrome because of hyperammonemia, hyperornithemia, and homocitrullinemia. So as ornithine is not transported back from cytoplasm to mitochondria, therefore in the mitochondria, carbamoyl phosphate combines with another basic amino acid that is lysine to form homocitrulline. So that is where your homocitrullinemia is coming from. Let me show you diagrammatically. This is your mitochondria, okay? Now what is happening? Your ornithine in cytoplasm, it cannot be transported into the mitochondria. Okay, so this here is blocked. This is not taking place because this transporter is defective. So what is happening? Your ornithine starts to accumulate hyperornithemia in the cytoplasm. Now in the mitochondria, what is happening? Your carbamoyl phosphate was supposed to combine with ornithine to form citrulline, but since the transporter is defective and the ornithine cannot reach mitochondria, so your carbamoyl phosphate in the mitochondria, it is combining with another amino acid, which is lysine. So when your carbamoyl phosphate combines with lysine, what is formed? Homocitrulline is formed. And this is what is contributing to homocitrullinemia and hyperammonemia we've already seen. So, as ornithine is not transported back from cytoplasm to mitochondria, therefore in the mitochondria, carbamoyl phosphate combines with another basic amino acid, lysine to form homocitrulline. So these were the distinguishing features of HH8 syndrome. It was due to defect in ornithine transporter. Now, what is in arginemia? What is happening? Your arginase is defective. But as you know that this is the last step in the urea cycle, your arginase enzyme is the last enzyme. So by the time arginine accumulates, both the nitrogen have already been abducted. That is, they are being abducted in the non-toxic form. Okay. So both nitrogen have already been abducted. Non-toxic form, that is. So which can be excreted in urine. So this is, therefore there is a least hyperammonemia. This is what is contributing to least hyperammonemia. So if you get an MCQ that among the following HH8 syndrome, arginosuccinic aciduria, or citrullinemia, and arginemia, which of the following shows all of them are urea cycle disorders, but which of the following has least hyperammonemia? So your answer will be arginemia. Why? Because this is the last enzyme in the cycle. So by the time the arginine accumulates, your two nitrogens have already been abducted. They have already been reduced to their non-toxic form. So therefore, they can be excreted in the urine and it contributes to least hyperammonemia. Now, clinical features of urea cycle, the classical triad of urea cycle. Okay, what is the classical triad of urea cycle? That is encephalopathy. Encephalopathy, we've already seen in the beginning of the lecture how I described you that ammonia is causing toxicity. What is encephalopathy? It is a broad term for any brain disease that alters your brain function and structure. Okay, so we've seen how the structure and function was being altered. Your ammonia was increased. When ammonia was increased, it was able to cross the blood-brain barrier in the brain. What was happening? It was being accepted by alpha ketoglutarate. So your alpha ketoglutarate was getting depleted, and since alpha ketoglutarate is an important subset of TCA, so what was being affected? TCA was being affected, so there was low energy levels, low ATP production. Ammonia, what was forming? It was forming glutamate. So your glutamate level was increasing, and when glutamate level increases, the glutamate, excess glutamate, it was on decarboxylation forming GABA. So your GABA was increasing, and GABA is a primary inhibitory neurotransmitter of the brain, so it causes CNS depression. Also, your glutamase was accepting ammonia to form what? Glutamine. So your glutamine levels were also increasing, and glutamine has the property, it is an osmotically active compound and has the property of attracting water. So it was leading to cerebral edema. Okay. So all these things were contributing to what? Encephalopathy. Hyperammonemia, we've already discussed. Then respiratory alkalosis is the last in the classical triad of urea cycle. How is respiratory alkalosis being contributed? The excess ammonia, it causes hyperventilation. Okay. So when there is excess ammonia, it leads to hyperventilation. Leading to what? So there is carbon dioxide, that means carbonic acid wash out. Okay. Wash out. So this contributes to respiratory alkalosis. So in urea cycle disorders, you have tachypnea, that is fast breathing. So that was all about urea cycle and the associated disorders of urea cycle. So with this, we come to the end of the class. Tomorrow we'll be talking about glycine, serine, and tryptophan metabolism. If you have any queries, you can ask. Thank you for watching. Okay. One more thing here. When we discussed ammonia toxicity, this. So this ammonia toxicity, all this was happening when there is excess ammonia, okay? When there is excess ammonia, okay? So little amount of ammonia is transported as ammonium ion, which is impermeable. So it is all fine until then. But when there is excess ammonia and that ammonia starts to accumulate, and that ammonia which is permeable to the blood-brain barrier crosses the blood-brain barrier and reaches the brain, that is when your toxicity of ammonia is manifested. Okay, otherwise in lower quantities it is transported as ammonium ion. Okay.